Hello students, Manjunath here. So in the previous class we had discussed about the particular chapter, that is sexual reproduction in flowering plants, and let us continue with the same part. Okay, the next one is pollination. What is pollination? Coming to the first question, that is the transfer of pollen grains to the stigmatic surface is called the pollination. The process of transfer of pollen grains from anther to the stigmatic surface. Okay. So coming to the question, that is what is pollination? Pollination is nothing but what? The process of transfer of pollen grains from the anthers to the stigmatic surface. So that is called pollination. And this particular mechanism will include different types, or the particular process of pollination will be taking place by means of different methods. Okay. Let us go through them one by one. So coming to the first one, okay, the pollination can be of the three different types, namely autogamy, geitonogamy and xenogamy. The first one is autogamy. So what exactly is meant by autogamy? So here the transfer of pollen grains will take place from the anther to the stigma of the same flower. That means it is as good as self pollination. Okay, it is nothing but what? Self pollination. What is self pollination? It's a type of pollination in which the transfer of pollen grains will take place from the anther to the stigma of the same flower. That itself is called as what? The autogamy, or which can also be called as self pollination. Generally, the autogamy or self pollination will be seen in the bisexual flower, as because for the transfer of pollen grains from the anther to the stigma of the same flower, it should be what? Bisexual. Okay, for the transfer of pollen grain from the anther to the stigma of the same flower, the flower should be bisexual. Bisexual means what? Both male and female sex organs should be present in the same flower. Okay, as in case of hibiscus, say for example, both male and female reproductive structures, okay, they will be present in the same flower. Okay, those type of flowers are called as what? Bisexual flowers. Okay, and you remember that there occur unisexual flowers also. So unisexual means they will be having either male reproductive part or else female reproductive part. Okay, either stamens they will be having or else they will be having pistils. Okay, remember that if they have got only stamens, then they are called as staminate flowers. Okay, if they have got only stamens, then they are called as what? Staminate flowers. And if they have got pistils only, then they are called as pistillate flowers. Okay, staminate flowers and pistillate flowers in the unisexual type. Okay, unisexual flowers ಅಲ್ಲಿ staminate ಮತ್ತು pistillate ಅಂತ ಎರಡು ಥರ ಬರ್ತಾವು. Staminate flowers ಅಂತಂದರೆ it will have only the male reproductive part, that is stamen. Okay, and if it has got only the female reproductive part, then that particular type of flower is called pistillate flower. Okay, so the autogamy will be seen in not the staminate or not the pistillate flower, but it is seen in bisexual flower. That means it will be seen in what? The flower which has got both the sex organs, that is male as well as female reproductive parts, which will be present in the same flower, right? So coming to the next one, that is autogamy or the self pollination is of two types. Okay, in autogamy or in self pollination there are two types. The first one is chasmogamy and second one is cleistogamy. Okay, the first one chasmogamy and second one is cleistogamy. So coming to the first one, that is chasmogamy. Chasmogamy is nothing but a type of autogamy or self pollination which is seen in the flowers, and these particular flowers, okay, are so special that they have got the anthers and the stigma lying very close to each other. That means chasmogamous flowers are those flowers in which the male and female reproductive parts are lying very close to each other, so that the pollination or self pollination will be confirmed very easily or will be taking place very easily. Okay, they will not be present very distant. Okay, in case of chasmogamous flowers, what happens? Male and female reproductive parts will lie very close to each other, right? So that is called as what? Chasmogamous flowers. And coming to the next one, that is cleistogamy or cleistogamous flowers. The flowers which show cleistogamy are called cleistogamous flowers. So it is a type of self pollination again, in which the self pollination will be confirmed in such a way that, okay, the flowers will not open at all. Okay, if the flowers do not open, okay, which will be bisexual in nature, then the anthers which will dehisce inside the flower itself and the pollen grains will be released on the particular part or female reproductive part called as stigmatic surface. Okay, if the flowers open, then only there will be chance of what? Missing of this particular self pollination. If the flower do not open at all, then self pollination will be confirmed very easily, right? So that type of flowers are called as chasmogamous flowers or chasmogamy, right? So next one, okay, this is about autogamy or self pollination, okay, and the types. Coming to the next one, that is geitonogamy. Okay, what is geitonogamy? It's a type of pollination in which the transfer of pollen grains takes place from anther to the stigma of another flower. Okay, here, see here, pollen grains will be transferred from one flower to another flower. Okay, the transfer of pollen grains will take place from anther of one flower to the stigma of another flower. But the point here is very important, that is, even though the transfer of pollen grain take place from one flower to different flower, both the flowers will belong to the same plant. Okay, that means it is also a type of pollination in which the cross pollination will be taking place, but what will happen? Between the flowers of the same plant. Okay, that is why geitonogamy, the transfer of pollen grains from anther to the stigma of another flower of the same plant. Okay, see here, although geitonogamy is functionally a cross pollination involving a pollinating agent, genetically it is similar to autogamy. Why it is genetically similar to autogamy? Because the transfer of pollen grains will be taking place, no doubt, from one flower to other flower, but both the flower, since they belong to the same plant, it will be genetically identical or genetically similar to that of what? Autogamy or self pollination. Is that clear? Because there is no chance of crossing between another plant or a different plant. Okay, the flowers, since they belong to the same plant, okay, what happens? Geitonogamy will act like self pollination itself, right? Coming to the next one, that is xenogamy. It is nothing but the transfer of pollen grains from anther to stigma of different plant. Okay, and this is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Okay, in case of xenogamy, okay, cross pollination will be seen. Okay, cross pollination means what? The transfer of pollen grains from one particular flowering plant to another flowering plant. Okay, to the from the anther of one particular flower, okay, and one particular flowering plant to the anther of another particular flowering plant, right. So that will be taking place in case of xenogamy, right. So coming to the next one, that is agents of pollination, okay, agents of pollination. Okay, coming to the agents of pollination. So, what exactly is meant by the agents of pollination or pollinating agents? See, dear friends, remember that for the transfer of pollen grains from a particular plant or from anther of a flower to the stigma of another flower, okay, or the same flower, it is not possible for the pollen grain to move freely. Why? Because the pollen grains are non-motile or immotile structures. Okay, for their transfer towards the stigmatic surface, they need some factors, whether it may be biotic or abiotic. That means the pollinating agents may be a living organism or it may be a non-living thing, that is abiotic factor. Okay, so that particular abiotic factor or the living organism which help for transfer of pollen grain from the anther to the stigmatic surface are called as agents of pollination or pollinating agents. Okay, I repeat the point. Do remember, agents of pollination or pollinating agents. These are nothing but the biotic or abiotic factors. Okay, biotic agents or abiotic factors which help for transfer of pollen grains from anther to the stigmatic surface. Okay, they help for transfer of pollen grains from anther to the stigmatic surface. Is that clear? Since they help for what? The transfer of pollen grains, they are called as pollinating agents. Okay, they are called as pollinating agents. And based upon the pollinating agents, the pollination can be divided into, or the pollination can be described into different types. Okay, based upon the pollinating agents, the pollination can be of different types, okay, as mentioned below. See, look at the first one, anemophily. Okay, the word phily, phily, phily means what? The transfer. Okay, anemophily means the transfer of pollen grains from anther to the stigmatic surface by means of a biotic factor, that is air or wind. Okay, anemophily is nothing but the type of pollination in which, okay, anemophily is nothing but the type of pollination in which the transfer of pollen grain from the anther to the stigmatic surface will take place with the help of the abiotic factor, that is wind or air, right. Say for example, in case of wheat and grass, this type of pollination can be seen. Okay, in case of wheat and grass, this type of pollination can be seen. And do remember, students, in case of anemophily, okay, the plants which show anemophily, say for example wheat and grass, they will not have brightly colored flowers. Okay, they will not have what? The brightly colored flowers. Why? Because there is no question of attraction. Okay, because the pollination is taking place with the help of wind or air, which is abiotic in nature. Okay, since the pollinating agent is abiotic, then there is no need of attraction. Okay, there is no need of what? Attracting the wind. That is why in these particular plants the flowers will not be brightly colored. Okay, and they may be even lacking what? The corolla or the accessory whorl, that is corolla or petals. Say for example, in case of wheat and grass, you could not see any brightly colored part, that is corolla you could not see. You can just see what? The spike. Okay, the type of inflorescence in case of wheat and grass. Okay, that is called as spike inflorescence. That you can see. But the thing is, here the colored flowers cannot be seen. Why? Because the brightly colored flowers will be present only in the plants where the pollination will be taking place with the help of insects, okay, or with the help of what? Biotic factors. But here, in case of anemophily, the pollination is taking place with the help of air or an abiotic factor. That is why the flowers will not be brightly colored. Right. So, coming to the next one, that is hydrophily. Hydrophily, by the name itself, you can easily recognize what is hydrophily. It's nothing but what? The type of pollination in which the pollinator or pollinating agent is what? Again, an abiotic factor called as water. If water is helping out for the transfer of pollen grains from stigma to the from anther to the stigmatic surface, then it is called as hydrophily. That means pollination with the help of what? Water. And this particular type of pollination by means of water will be seen in aquatic plants. Okay, you already can know that, or you already can get it. Okay, because pollination by water will be taking place in the aquatic plants itself. And which all the aquatic plants? Examples are Vallisneria and Hydrilla. So, Vallisneria and Hydrilla are the two aquatic plants in which the pollination or the pollinating agent is water. Pollination will be taking place with the help of water, right? So, coming to the next one, that is zoophily. Okay, zoophily. Okay, phily means pollination or the transfer of pollen grains. And what about zoo? Zoo is nothing but what? Animals. Okay, so zoophily is nothing but the pollination by means of animals. Okay, it's a type of pollination where transfer of pollen grains will take place with the help of animals. Okay, and many different types of animals will help for, okay, the particular process of pollination. Okay, whether they may be very small sized animals such as the insects, or it may be any cattle. Okay, so they directly or indirectly will help for transfer of pollen grains from anther to the stigmatic surface of the flowers. That is why zoophily is most important, and at present, at present, more than 90% of the food crops are resulted with the help of what? The zoophily itself. Okay? Particularly, the honeybees are playing a major role in the zoophily. Okay? Because honeybees are the pollinating agents. Okay? Honeybees are the pollinating agents. Okay, most of the flowers are pollinated with the help of honeybees itself. Most of the flowers, particularly more than 90% of the flowers of present day are pollinated by the honeybees. Okay, so there in the scientists say that without honeybees, okay, without honeybees no food crops can be produced. Okay, without honeybees no food crops can be produced. According to the scientists, the food crops are produced only by means of pollination through the what? The honeybees. Okay, exceptionally, rarely, only few plants can be produced without the honeybees, where the pollination will be taking place by the wind. Right. So, next one, that is chiropterophily. Okay. So, before going to chiropterophily, okay, zoophily, I'll just mention one point here. The zoophily, or the plants which show zoophily, they will be having, okay, brightly colored flowers. Okay, the plants which show zoophily will be having brightly colored flowers. And another important point is their stigmatic surface will be having high amount of nectar. Then third point is these particular flowers, okay, will be having the sticky type of what? The pollen grains. That means pollen grains of this particular plant which are pollinated by the animals will have sticky pollen grains, okay, so that it can stick to the surface of the animals, okay, which will be helpful for what? The pollination. Coming to the next one, chiropterophily. Okay, chiropterophily. What is chiropterophily? Very simple. It's a type of pollination which will be resulted by means of what? The bats. Okay, you know already the bats. The bats will help for, or the bats will directly or indirectly help for transfer of the pollen grains in some of the plants. Okay, the transfer of pollen grains from stigma, from anther to the stigma of the particular flower will take place with the help of bats, so that the bats will act as pollinating agents, and this type of pollination with the help of bats is called as chiropterophily, right? The next one, entomophily. Coming to the next one, that is entomophily. What is entomophily? It's a type of, do remember, entomophily is a type of pollination again, where in which the pollinating agent will be an insect. Okay, entomophily is the particular type of pollination in which the pollinating agent, okay, or agent of pollination will be an insect. Okay, the pollinating agent will be an insect, and these flowers which are pollinated by the insects will have the special characters such as brightly colored flowers. Second, the sticky type of what? Anthers or sticky type of pollen grains. Then third one is high nectar producing stigmatic surface. For the purpose of nectar, the insect will visit the plant and insect will visit the particular flower, okay, during which the pollen grains which are sticky will be transferred to what? The stigmatic surface, because the sticky pollen grains will get adhered or attached to the surface of the body of these particular insect. Okay. After that, when the insect will move towards the stigmatic surface, it will unload what? That particular pollen grains on the stigmatic surface. At the same time, in case of entomophily, the insects are also given with what? The particular prize, okay, for the purpose of helping the plant to get pollinated. Okay, tribute will be given for the insect in the form of nectar and pollen, okay, which they usually use for as their food. Clear? So like this, the pollination is an important mechanism by which the pollen grains are transferred from the anther to the stigmatic surface. Okay, different types of pollination and different types of pollinating agents are there, as I have explained now, right? And in these, the very important point is, what are the characters of entomophilous flowers? Okay, the characters of entomophilous flowers are very, very important. Okay, and the characters of anemophilous flowers. Okay, anemophilous flowers will not be brightly colored, but entomophilous flowers are brightly colored, right? Like this, the pollination will be taking place with the help of different types of pollinating agents, right? Coming to the next one, outbreeding devices. So what is outbreeding device? Very simple. Remember friends, due to continuous inbreeding or continuous self-pollination, okay, self-pollination can also be termed as inbreeding, okay. So due to continuous self-pollination, there occurs a major problem called as inbreeding depression, okay. Continuous self-pollination ಆಗೋದ್ರಿಂದ ಏನಾಗುತ್ತೆ ಅಂತಂದರೆ inbreeding depression ಆಗುತ್ತೆ. What is inbreeding depression? It is nothing but the condition in which, due to continuous inbreeding or continuous self-pollination, same genes will be present or same genes will get accumulated. Okay, no other variations will be seen. Okay, due to that, what will happen? The plant will not have any improvement in its yield or in its character. Okay, That is why what happens? The inbreeding depression will be taking place. Is that clear? So to avoid the inbreeding depression, okay, to avoid the inbreeding depression, some flowering plants have developed some of the mechanisms, okay, or they are also called as some devices to avoid the inbreeding depression. Okay, some plants have developed some mechanisms or devices to discourage the self pollination or to avoid the self pollination. Some of the plants have developed their own mechanisms. Okay, so that itself are called as what? The outbreeding devices. What are outbreeding devices? These are the devices or mechanisms by which some of the organisms, some of the plants have developed what? Some devices to discourage self pollination. Okay, the outbreeding devices are as follows. The first one is receptivity will be not synchronized. That means in some species, in some plant species, what will happen? The pollen release and stigma receptivity are not synchronized. That means pollen grains will be released at one particular time during which the stigmatic surface will not be receptive for the pollen grains. Okay? The time period will be varied. If the pollen grains are released at one particular time, okay, during that time stigma will not be receptive. Okay, so that even though the transfer of pollen grains take place on the stigmatic surface, it will not be receptive. Since it is not receptive, the pollen grain which falls on the stigmatic surface cannot lead to what? Fertilization. It cannot lead to fertilization. That is how they will avoid the self pollination. First one, that is the pollen release and stigma receptivity will be not synchronized. ಅಂದರೆ ಬೇರೆ ಬೇರೆ ಟೈಮಿಗೆ ಆಗ್ತವೆ. ಏನಪ್ಪ ಅಂತಂದರೆ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಒಂದು ಟೈಮಿಗೆ ರಿಲೀಸ್ ಆದರೆ ಆ ಟೈಮಲ್ಲಿ ರಿಸೆಪ್ಟಿವಿಟಿ ಆಫ್ ದ ಸ್ಟಿಗ್ಮಾ ಇರುವುದಿಲ್ಲ. ರಿಸೆಪ್ಟಿವಿಟಿ ಆಫ್ ದ ಸ್ಟಿಗ್ಮಾ ಇದ್ದಾಗ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರಿಲೀಸ್ ಆಗಿರಲ್ಲ. So that if both of them are released at a different time period, then there will be no self pollination. This is how they will avoid what? The self pollination, right? And they will encourage the cross pollination. In second one, okay, in some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. That means in case of some plants, the pollen producing structure, that is anther, and the receptive structure, that is stigma, both of them are produced at a distant places. Okay, at distant places, so that the transfer of pollen grains will not be taking place very easily, right? So this is another method of avoiding what? The self pollination, right? The third one is the self incompatibility. Okay, the third device is to prevent the inbreeding is self incompatibility. What is self incompatibility? It is nothing but what? The type of what? Outbreeding device in which, okay, the type of outbreeding device in which what will happen? The same plants flowering, okay, particularly the anther and stigma, they will not be compatible with each other. Okay, even though pollination take place, okay, even though pollination take place, it will not lead to the fertilization. Why? Because they are incompatible. As if you apply your home key or your bike key to the next one or to any other bikes, that will not be compatible. Okay, or if you put the mobile cover of one particular mobile to another which is different, okay, it will not be compatible. Like the same here, what will happen? When the pollen grains fall on the stigmatic surface, that particular pollen grain will be incompatible with the stigmatic surface. The compatibility will not be matching. If the compatibility is not matching, then what will happen? It will lead to cross pollination or it will avoid the self pollination. Compatibility irudilla. Clear? So next one, Another device, another device to prevent the self pollination is unisexual flowers. Remember that. What are unisexual flowers? These are the flowers which will be having the reproductive structure of only one gender. That means either it may have the reproductive structure of male or it will be having the female reproductive structure. Okay. If it has got the male reproductive structure only, then that type of flower is called as what? Staminate flower. Okay, it is called as staminate flower. And if it has got the pistil or female reproductive part only, then it is called what? The pistillate flower. Okay, like that, the flowers will be avoiding for self pollination by means of being unisexual. Okay, being what? Unisexual, right? In case of hibiscus, you can see it is bisexual, but in some flowers what will happen? Unisexual flowers can be seen. Okay, in some plants unisexuality can be seen. Coming to the next one, that is, in several species such as papaya, male and female flowers are present on different plant itself. That means here in the fourth one, unisexual flowers, the flowers will be unisexual but they may be present in the same plant. But in some cases what will happen? The complete plant itself will be what? Having only male flowers or only female flowers. That means the plant completely itself will act as either male plant or female plant. So examples are papaya plant. In case of papaya plant, male and female flowers are present on different plants completely. Okay, that is, each plant is either male or female. Okay, that is called as dioecious. Okay, dioecious nature anta karitivi. Okay, whether the plant will have either male reproductive part or else female reproductive part. Is that clear? So that is how it will be. So let us move to the next one, that is pollen pistil interaction. Okay, what is pollen pistil interaction? See, soon after the pollination, what will happen? Soon after the transfer of pollen grain from anther to the stigmatic surface, okay, when the pollen grain comes in contact with the stigmatic surface, what happens? Okay, those particular processes are termed as what? Pollen pistil interaction. Okay, and what happens after the appearance of pollen grain on the surface of what? The stigmatic surface. So the ability of pistil to recognize the pollen followed by its acceptance or rejection is termed as pollen pistil interaction. That means when a particular pollen grain falls on the surface of a stigma, then what happens? The stigma will first check whether the pollen grain is of its own kind or not, whether to take the pollen grain or whether to allow the pollen grain to germinate, or whether to allow the pollen grain to germinate the pollen tube and fertilize or not, whether it should accept the pollen grain or it should reject the pollen grain. That will be decided by what? This particular stigmatic surface. And this interaction between the pollen grain and the stigmatic surface itself is termed as pollen pistil interaction. The pistil has ability to recognize the pollen. Do remember? Pistil has, see here, second point. The pistil has ability to recognize the pollen where whether it is right type or compatible or of the wrong type or incompatible. That means when the pollen grain falls on the surface of the pistil, it will first recognize whether the pollen grain is of its own kind or not. If it is of its own kind and if it is capable of fertilizing, then it will accept. Okay? If it is compatible, then it will accept. If it is not compatible, then the pollen grain will be rejected by what? The stigmatic surface. Okay? Only if it is compatible, then it will be accepted, right? So that particular process is termed as what? Pollen pistil interaction. Okay, that particular process is termed as pollen pistil interaction. See, if the pollen which is appeared or assumed on the surface of the stigma, if it is right type or if it is compatible, then what will happen? The pistil accepts the pollen grain. Okay, the pistil accepts the pollen grain and promotes the post pollination events. Okay, if it is compatible, if it is acceptable, if it is of its own kind, then what will happen? The pistil will accept the pollen grain and it will promote what? The post pollination event. That means it will promote the further events that take place after pollination. Okay, what are the further events? Very simple: the development of the pollen tube and fertilization. Okay, that itself is called as the pollen germination and fertilization. Is that clear? Next one, artificial hybridization. Okay, what is artificial hybridization? Okay, it's a type of, okay, it is a type of pollination that is done artificially. Okay, it's a type of pollination which is done artificially. That means even the human beings, okay, even the scientists or the human beings can do the pollination artificially by means of very simple technique. Take two flowers of opposite sex, or if you want to cross any two plants, just you have to collect the flowers, okay, freshly sprouted or freshly blossomed flowers, and from that what you have to do, you have to choose male and female. Okay, in the male flowers you have to remove the female one, and in the female flowers you have to remove the male reproductive part. Then what happens? Soon after the dehiscence of the anther, you have to transfer those pollen grains on the stigmatic surface of the plant you which want to get fertilized. So that is artificial hybridization. By means of artificial hybridization, we have improved and the farmers have developed lot many number of improved varieties of the crops. Okay, the food crops are improved, okay, or improved food crops are developed by the farmers by means of artificial hybridization so many times. Coming to the next one, that is emasculation and bagging. What is emasculation? Very simple thing. Students, do remember, emasculation is a very important step in case of artificial hybridization. I told you, when you select any two plant, you have to select one male and one female. And if you want to make a particular plant as a female, in case if it is bisexual, then what you have to do? You have to take off the male reproductive part. Like the same, in case of emasculation, what we are going to do, the process, okay, emasculation, we are going to remove the anthers. Okay, we are going to remove the anthers from the floral bud before the dehiscence. Okay, do remember what is emasculation? It is the type of, or it is the process in which the removal of anthers will be done. Okay, removal of anthers will be done by means of what? Cutting them before their dehiscence. Okay, removal of anthers before their dehiscence from a bisexual flower is called what? Emasculation. I repeat the point. Removal of anthers from a bisexual flower before their dehiscence is called emasculation. Okay, why the emasculation is done? Very simple. If the emasculation or removal of anther is not done before their dehiscence, what will happen? Immediately, soon after their dehiscence, they will transfer their pollen grain to the stigma of the same flower. Then what will happen? Hybridization cannot be done because immediately pollination will be taking place there. Okay, so for hybridization, you have to select a flower which has not undergone any of a what? Pollination. Okay, that is why we are going to select the flower and it should be in the form of what? Bud. And that flower bud which will be selected. From that flower bud, what we have to do, we have to take out the anthers before their dehiscence or before the release of pollen grains, which is called as emasculation. And what about bagging? The emasculated flower will not be left as it is or should not be left as it is openly to the air. It should be tied or it should be covered with a plastic bag. Okay, it should be covered with a plastic bag with minute pores so that aeration will be taking place. When the particular emasculated flower is covered by, okay, the plastic bag, it will confirms no other pollen grain will be falling on that particular stigma. ಅಂದರೆ ಈಗ ಒಂದ್ಸಲ ನಾವು ಏನಾದರೂ ಎಮಾಸ್ಕುಲೇಷನ್ ಮಾಡಿದಂಥ ಫ್ಲವರನ್ನು ತಗೊಂಡ್ರೆ ಆ ಫ್ಲವರಿಗೆ ಬೇರೆ ಆದಂಥ ಯಾವುದಾದ್ರೂ ಪೋಲನ್ ಗ್ರೈನ್ ಬಂದು ಪಾಲಿನೇಟ್ ಮಾಡಬಾರ್ದು ಅನ್ನೋ ಒಂದು ಉದ್ದೇಶಕ್ಕೆ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕವರ್ ದ ಸೇಮ್ ಸ್ಟಿಗ್ಮೆಟಿಕ್ ಸರ್ಫೇಸ್ ವಿತ್ ದ ಹೆಲ್ಪ್ ಆಫ್ ವಾಟ್, ಅ ಪ್ಲಾಸ್ಟಿಕ್ ಬ್ಯಾಗ್. ಅದಕ್ಕೆ ಏನಂತ ಕರಿತೀವಿ, ಬ್ಯಾಗಿಂಗ್ ಅಂತ ಕರಿತೀವಿ. ಕ್ಲಿಯರ್? ಸೊ let us go to the next one, that is double fertilization. What do you mean by double fertilization? Very simple thing. Fertilization occurs twice. Okay, in case of angiosperms, fertilization occurs twice, and that itself is known as double fertilization. Okay, that itself is known as what? Double fertilization. So let us go to the double fertilization. See, soon after the pollination, in case of angiosperms, what happens? When the pollen grain comes and approaches the surface of the stigma, then the pollen pistil interaction will be initiated. Soon after pollen pistil interaction, when the stigmatic surface recognizes, okay, when the stigmatic surface recognizes what? The pollen grain as if its compatible nature or as if its own, then it allows the particular pollen grain to germinate. When the particular pollen grain starts germinating, it will start producing a elongated tube-like structure called as pollen tube. And through this pollen tube, the male gametes will be released. Okay, through this pollen tube, what will happen? Male gametes will be released. How many male gametes will be released? Remember that 2 male gametes will be released through the pollen tube. Okay, I repeat from the beginning, do remember. When the pollen grains falls on the surface of the stigma, pollen pistil interaction will take place. And when the stigmatic surface recognizes the pollen grain, it allows the particular pollen grain to germinate. After germination, the pollen grain will start producing what? A pollen tube. And this pollen tube will enter inside the stigmatic surface and it will enter to what? The ovarian region by passing through the what? The elongated part of the ovary or elongated part of the pistil, that is called style. From the surface, the pollen tube will be germinated, or from the surface, the pollen tube will be formed. And through this pollen tube which is developed, the male gametes will be transferred. Okay, and these male gametes will be 2 in number. Okay, do remember, the male gametes which are released in the pollen tube, it will not be single, it will be 2. That means 2 pollen, 2 what? The microspores or 2 particular nuclei will be released, or 2 gametes will be released. Okay, and these 2 gametes which are released in the pollen tube, okay, they will ultimately reach the embryo sac. Okay, they will ultimately reach what? The embryo sac or the ovule. Okay, and here you have already studied, okay, in the previous part only I have discussed about what? The structure of ovule and the embryo sac. When the pollen tube reaches the micropylar end, okay, when the pollen tube reaches the micropylar end, it will approach or it will reach what? 3 particular cells there. First one is egg cell, and other 2 which are present along with the egg cell are called as what? Synergids. And these synergids, I have told you, they will have finger-like projections. Okay, They will have what? Finger like projections. And those finger like projections are called as, okay, those finger like projections are called as what? Filiform apparatus. Okay. And when the pollen tube approaches this particular micropylar end and the synergids, the filiform apparatus which is present there, it will guide the male gametes towards egg cell and another male gamete towards what? The polar nuclei. Okay. Then what will happen? One male gamete, okay, which has entered inside will reach and fertilize the egg cell, and another male gamete will fertilize the polar nuclei. Okay. So one gamete which is entered here, it will fertilize or it will fuse with the egg cell, and other gamete which has entered inside, it will fuse with what? The polar nuclei. Do remember that when the first or primary nuclei, okay, primary nucleus, okay, it fuses with the egg cell, it will be called as what? Syngamy or fertilization. When the first male gamete, when the first male gamete enters inside, okay, it will fuse with the egg cell, and that will be called as what? Syngamy. Okay, when it fuses with the egg cell, it will be called as syngamy or fertilization. And other male gamete, okay, will be fused with what? The polar nuclei. The polar nuclei are two in number, and with these two nuclei, another male gamete or male nuclei will be fused. That is why the fusion of three nuclei will take place. That itself is called as triple fusion. Okay. Here the word double fertilization means what? Fertilization occurs twice. That means one male gamete will fertilize the egg cell, that is first fertilization. Second male gamete will fertilize what? Or fuse with what? The polar nuclei. That means fusion of male and female gamete will take place twice. That itself is called as double fertilization. Okay. And here in the polar nuclei, they are already two in number, and along with that, one particular male gamete will be fused. That will confirms what? Triple fusion. By means of triple fusion, what will be developed? The primary endosperm nucleus will be developed. Okay. Triploid primary endosperm nucleus. See the fourth point. The other male gamete move towards what? Two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus. Why the primary endosperm nucleus is called triploid? Because it is formed by fusion of three nuclei. That is why it is called as what? Triploid primary endosperm nucleus. And what this primary endosperm nucleus will do further, or what it will become further? The primary endosperm nucleus in further or in future will be acting as nutritive tissue, or will develop into nutritive tissue for the developing embryo. And here the first syngamy which has taken place with the egg cell, okay, egg cell ಮತ್ತೆ first ಯಾವುದು gamete fuse ಆಗಿರುತ್ತಲ್ಲ, so this particular point will lead to what? Zygote formation. So the zygote which is formed here will develop into an embryo, and the primary endosperm nucleus will form the endosperm, which is the nutritive tissue for the developing embryo. Is that clear? So that itself is what? Double fertilization and triple fusion. By means of double fertilization and triple fusion, what all the structures have been produced here? First one, zygote is produced by means of syngamy or else by means of fertilization. Second one, the primary endosperm nucleus will be produced, and this primary endosperm nucleus will lead to the formation of endosperm. And very important point here is primary endosperm nucleus is triploid in nature. Okay, the primary endosperm nucleus will be triploid in nature. Clear? Okay. This particular process of fusion of two male gametes with one egg cell and two polar nuclei is called double fertilization and triple fusion. Clear? Okay, very, very important for your NEET exam point of view, right? Coming to the next one, that is post fertilization events. Okay, post fertilization events. What do you mean by post fertilization events? Nothing but the events that occur soon after fertilization. Okay, what are the processes that takes place after fertilization are called as post fertilization events. Okay, after the fertilization, what all the things will happen? I have told you. Following double fertilization, okay, the primary endosperm nucleus will be formed, and that primary endosperm nucleus will develop into endosperm, which is a nutritive tissue for the developing embryo. Okay, that is first point. Then second one is what? The embryo which has formed, it will start getting development and it will undergo maturation. Okay, next the ovules develop into what? Seeds. Okay, the ovule will develop into seed and ovary will develop into fruit. Okay, very important point, students. Do remember, after fertilization, okay, after fertilization, ovules will develop into seeds and ovary will develop into fruit. Okay, ovules will develop into seeds and ovary will develop into fruit, right? And what about primary endosperm nucleus? The primary endosperm nucleus will ultimately form the nutritive tissue called as endosperm. Okay, and look at the last point. Endosperm may be either completely consumed by the developing embryo before the seed maturation, or it may persist in the mature seed. Very important point here. The endosperm which is formed may be completely consumed by the embryo, or it may even retained. Okay, in case of pea and in case of groundnut and beans, it will be completely consumed by the developing embryo. But in case of castor and coconut, okay, the endosperm will not be completely consumed, but it will be retained. So in case of coconut, you might have seen very oftenly, okay, coconut milk, what you call it as, so that itself is nothing but what? It is the persistent endosperm. That itself is nothing but persistent endosperm. Okay, and in particularly in the coconut, you can see two different types of endosperm. One is cellular endosperm and second one is what? The liquid endosperm. Okay, cellular endosperm and liquid endosperm. That liquid endosperm is in particular called as free nuclear endosperm. Okay, it is called as what? Free nuclear endosperm. Okay, so that is very, very important to remember here, right? So I repeat the post fertilization structures and event. Very importantly, you have to remember this one. See friends, following fertilization, that means soon after fertilization, okay, what will happen? Two major events will take place. First one is by means of syngamy or fertilization, what will happen? The zygote development will take place, and that zygote which is unicellular will develop into multicellular embryo. Okay, embryogenesis will take place. Okay, and during the development of this embryo, it needs what? The food material or else the nourishment, and this nourishment was provided by, this nourishment which is required by the zygote for the embryogenesis will be given by the nutritive tissue which is developed by means of what? Primary endosperm nucleus. Okay, the endosperm will be formed, and what is endosperm? It is the nutritive tissue for the developing embryo. This nutritive tissue for the developing embryo will be produced by or will be given by what? The primary endosperm nucleus. Okay, or will be developed by primary endosperm nucleus. Okay, in one sentence, if you want to say, the primary endosperm nucleus will develop into endosperm to give rise to what? To give rise to the nutritive tissue. Okay, and it will ultimately help for nourishment of the zygote. Okay, and this zygote with the help of what? The endosperm will develop into an embryo. Okay, next one, The ovule, okay, ovule after fertilization will develop into seed, and the ovary after fertilization will develop into fruit. Clear? And the endosperm which is produced, it may be completely consumed or it may be retained. In case of pea plant, in case of groundnut, and in case of beans, say for example, the endosperm which is produced will be completely consumed. But in case of what? The castor and coconut, the endosperm will be remaining or else it will be persistent. Okay, it may be remaining as it is. That means part of the endosperm will be consumed and part of it will be remaining. Okay, and in the coconut there are two types of endosperms you can see. First one is the endosperm which is in the form of liquid, that is liquid endosperm or else free nuclear endosperm. Okay, which you call it as what? Coconut milk. And ಅದೇ coconut milk ಒಳಗಡೆ ಏನಿರ್ತದೆ, ಅದೇ coconut ಒಳಗಡೆ you can see the white colored cellular matter. Okay, the white colored cellular matter of the coconut is nothing but what? Cellular endosperm. Okay, the liquid part is called as liquid endosperm and white cellular matter is called as what? Cellular endosperm. Right. This is how different types of endosperms can be produced, and in the next class we shall deal with what? The embryo which are formed. Right. Thank you.